Good afternoon, Professor. This is my first video format test, and what this is over is um how will water react with the DNA and this protein once it's completed, ready to send out of the cell. And from my understanding, DNA is soluble in water, which means it can dissolve in water, and it can also influence how DNA takes shape and interacts with other molecules. Question number two. What kind of bonds will be present and why those particular bonds between the A strands of the DNA molecule between the nitrogenous bases such as adenine and thymine. And the answer to that is the bonds that will be present in the nitrogenous bases will be hydrogen bonds. The reason these bonds will be present is because hydrogen bonds are easier to break, allowing for DNA copying. B, what kind of bonds will be present in the backbone of the DNA molecules, the sugars and the phosphate that hold the bases firmly to the DNA strand? The answer to that question is the sugars and the phosphates are held together by covalent bonds. These particular bonds will be present because they are stronger than hydrogen bonds, making the covalent bond crucial for the backbone of the DNA molecule. And what kind of bonds will be present in the molecules of protein, the amino acids? A, the link that is made between the amino acids is by a peptide covalent bond. And what will happen to the DNA and protein if the pH of the cell were to suddenly drop to 2.0? And when a DNA molecule drops to the level of 2.0 or becomes too acidic, the DNA molecule is prone to destabilization and is therefore liable to depurination due to the acidity of the molecule at that level.